In the white sand of New Mexico, in sediment that has been sealed and dated to at least 21,000 years ago, someone left footprints, and they should not have been there. The dates were repeated across multiple excavation areas, not a single anomalous result, but a consistent pattern. The sediment layers told the same story everywhere they were tested. The site spans over a kilometer, hundreds of prints from dozens of people, preserved in multiple layers representing centuries of visits. The prints were not metaphorical, they were physical. Actual human feet pressed into soft mud beside an ancient lake, preserved when the sediment hardened into stone. Toes, arches, heels. The unmistakable mark of people walking. The stratigraphy was unambiguous. The footprints were sealed between layers of datable material. Seeds above, seeds below, both returning the same date range. The prints could not have been intruded from a later period. The dates, 21 to 23,000 years before present. Radiocarbon dated from seeds embedded in the same sediment layers as the footprints. The seeds grew in the lake that once covered the area, deposited in the same mud as the footprints. Date the seeds, date the steps. And 21,000 years ago, according to every textbook published before 2021, there were no humans in the Americas. The dominant theory, Clovis first, held that humans entered the Americas 13,000 years ago via the Bering land bridge, the Clovis culture, named for spear points found in New Mexico, represented the first Americans. Clovis appeared suddenly, spread rapidly, and vanished within 3 centuries. The narrative was compelling and dominated archaeology for 70 years. Not all early American stone tools were Clovis. The Western stem tradition, found along the Pacific coast, was contemporary with or earlier than Clovis and represented a separate technological lineage, consistent with the coastal migration route. The original Clovis site at Blackwater Draw was discovered in the 1930s alongside mammoth bones. The association between the distinctive points and megafauna hunting defined the Clovis image. Big game hunters crossing the land bridge. The Clovis point was a masterpiece of stone technology, appearing across North America in a narrow window 13,000 years ago. The uniformity suggested a single migration. One people, one arrival. The Clovis model was elegant, simple, and wrong. The first crack came from the tip of South America. Tom Delahaye excavated Monte Verde in Chile, 14,500 years old, over 1,000 years before Clovis. Monte Verde preserved organic materials that almost never survive. Wooden tools, plant remains, even fragments of human-modified seaweed. The site was not a temporary camp. It was a settlement, occupied by people who knew the local plants and animals intimately. The Galt site in Texas produced artifacts from layers beneath the Clovis level, older technology stratified below the supposedly first culture. The stratigraphic evidence was as clear as a layer cake. If people were in Chile 14,500 years ago, they must have entered the Americas thousands of years earlier. Walking from Beringia to Patagonia takes time. Monte Verde was not a hunting camp. It was a village, with distinct living areas, a communal hearth, and stored food. The people who lived there were not arriving. They were settled. Wild potato skins preserved in the peat, the oldest evidence of potato use in the world. The people at Monte Verde knew the local plants intimately. That kind of knowledge takes generations to develop. Clovis defenders fought for decades, questioning, dating, stratigraphy, artifacts. Careers were built on Clovis first, and any challenge was heresy. But the evidence kept coming, from site after site, continent to continent, each one pushing the date back further. The Paisley Caves in Oregon produced stone tools and dried human coprolites, ancient feces, dating to 14,500 years ago, and the coprolites contained something the stones could not provide. Pollen cores from former Beringian lakes showed that the land bridge supported productive grassland ecosystems, capable of sustaining large herbivore populations, and the humans who hunted them. DNA, human mitochondrial DNA, extracted from desiccated feces left by people who lived in those caves over 1,000 years before the first Clovis point was made. The DNA belonged to haplogroup, a lineage still found in indigenous American populations today. The Paisley Caves had been known to archaeologists for decades, but it was the combination of datable organic material and recoverable DNA that made them revolutionary. The DNA confirmed what the archaeology suggested. These were not random visitors. They were the ancestors of the people who would populate the entire Western Hemisphere. Genetic studies of modern indigenous Americans had already identified 5 founding mitochondrial lineages, all tracing back to Northeast Asian source populations. The question was not where they came from. It was when they arrived. During the last glacial maximum, sea levels dropped over 100 meters, exposing Beringia, not a narrow isthmus, but a subcontinent over 1,000 kilometers wide, covered in grassland. Cactus Hill in Virginia produced stone tools from layers, dated to approximately 18,000 years ago. A pre-Clovis occupation on the Atlantic coast, suggesting early Americans had already spread across the entire continent. Beringia was not a frozen wasteland. Pollen records and insect remains show it was a productive steppe grassland, home to mammoths, horses, bison, and the predators that hunted them. Humans fit naturally into this ecosystem. The people who lived on Beringia were not migrants in the modern sense. They were residents of a subcontinent that happened to connect 2 others. When climate changed and routes south opened, some of them moved. People could have crossed at any point, but 2 massive ice sheets covered most of Canada, potentially blocking the route south. The route debate continues, but the timing debate is settled. However they arrived, humans were in the Americas at least 21,000 years ago. The footprints prove it. The DNA supports it. The argument now is about pathways, not dates. The ice-free corridor between the 2 sheets did not open until approximately 14,000 years ago. Too late to explain how people reached Monteverde by 14,500 years ago, or White Sands by 21,000. The alternative, the Pacific coastal route, following the kelp-rich shoreline south, bypassing the ice sheets by boat or on foot. The terrestrial pollen test was decisive. Pollen grains blown in from land plants cannot absorb ancient lake carbon. When their dates matched the aquatic seed dates, the hard water objection collapsed. The kelp highway hypothesis. Early Americans followed the productive coastline. Fish, shellfish, marine mammals. South from Beringia to Oregon and beyond. The problem: most Ice Age coastlines are now under 30 to 100 meters of water. The coastal camps are submerged. The kelp highway was one of the most productive marine ecosystems on Earth. Dense beds of giant kelp supporting fish, shellfish, sea mammals, and seabirds. Coastal travelers would have had reliable food sources along the entire Pacific shore. The absence of coastal evidence is not evidence of absence. It is evidence of submersion. The archaeological record of the coastal migration may be permanently underwater. And then came White Sands. The 2021 publication in Science reported footprints dated to 21 to 23,000 years ago, during the last glacial maximum itself, when the ice sheets were at their greatest extent. The footprints showed adults and children walking together, not a hunting party but a community. The smallest prints belonged to children as young as 3 or 4. These were families, living and walking in a landscape that the textbooks said was empty. The new textbooks are already being written. The revised timeline places initial entry into the Americas at least 20,000 years ago, potentially earlier, with the Clovis culture representing not the first Americans but the first highly visible archaeological culture. The change was not gradual. It was a paradigm shift, one that took decades of evidence to achieve against institutional resistance. The presence of children was significant. A scouting expedition might leave adult prints only. Families with young children suggest a settled population, people who lived here, not people passing through. The trackways also preserved animal prints, mammoth, giant ground sloth, and camelid tracks overlapping with the human footprints. People and megafauna were sharing the same landscape at the same time. Gait analysis of the footprints estimated walking speeds, body weights, and even the likely height of the individuals. The data was consistent with a population adapted to this landscape, comfortable, unhurried, at home. The paper was peer-reviewed, multi-author, and supported by multiple independent dating methods. It was not a fringe claim. It was published in one of the world's two most prestigious scientific journals. But the radiocarbon dates were immediately challenged because radiocarbon dating of aquatic plants can be affected by the hard water effect, where dissolved ancient carbon in lake water makes organic material appear older than it actually is. If the rupia seeds had absorbed ancient carbon from the lake water, the footprints could be thousands of years younger than reported, potentially falling within the already accepted pre-Clovis window, rather than representing a dramatically earlier arrival. Each ancient genome extracted from the Americas adds resolution to the founding story, confirming the Northeast Asian origin, refining the divergence dates, and mapping the routes of dispersal across two continents. The team responded with two more methods. OSL dating of quartz confirmed the radiocarbon results. Radiocarbon on terrestrial pollen, unaffected by hard water, confirmed the same range. The convergence of three independent methods was the key. Any one method can be challenged. Three methods returning the same answer from the same sediment is a different category of evidence. Three methods, three independent laboratories, the same answer. The footprints were real, and they were old. And across the Americas, ancient DNA was telling the same story. The founding population split from their Northeast Asian relatives not 13,000 years ago, but at least 20,000, possibly earlier. Molecular clock estimates placed the founding population split at 20 to 25,000 years ago, matching white sands almost exactly. The upward Sunday River infant from central Alaska, 11,500 years old, carried a previously unknown founding lineage called ancient Beringian, representing a population that stayed in the north while others dispersed south. Genetic continuity is remarkable. The same 5 founding lineages that entered over 20,000 years ago are still the most common in indigenous communities today. Multiple independent genetic studies using different statistical models converge on the same founding date, 22 to 25,000 years ago. The genetics and the footprints tell the same story. The genetics and the footprints agreed. The archaeological sites agreed. The only thing that disagreed was the textbook. The Anzac I child, the only Clovis-associated burial with recovered DNA, was genetically ancestral to all modern southern Native American populations. The Clovis people were not a separate migration. They were descendants of the same founding population that arrived thousands of years earlier. A 12,600-year-old child buried with Clovis tools in Montana carried the same founding lineages as people who left footprints in New Mexico 10,000 years earlier. The genetic thread was continuous. The Clovis first model dominated American archaeology for over 70 years. It was taught as fact in every university. It appeared in every textbook, and it was defended with a ferocity that went beyond scientific disagreement. Archaeologists who proposed pre-Clovis dates were marginalized and denied publication. The Clovis police enforced consensus with institutional power. Meadowcroft Rock Shelter in Pennsylvania produced dates exceeding 16,000 years in the 1970s. The excavator, James Adovasio, spent decades defending his findings against accusations of contamination and misinterpretation. He was eventually vindicated. Adovasio excavated Meadowcroft with meticulous stratigraphic control, documenting every layer, every artifact, every radiocarbon sample. His methods were impeccable. The attacks on his work were professional, not scientific. 21,000 years ago, the same stars shone over this landscape. The same sand recorded the same kinds of footprints. The people who walked here looked up at the same sky and left the only evidence that would survive to prove they existed. The stone tools from the deepest layers at Meadowcroft looked nothing like Clovis points. They were smaller, differently shaped, and made with different techniques. evidence of a separate and earlier technological tradition. The resistance was not about evidence. It was about careers and the reluctance to abandon a framework that structured an entire field. The pre-Clovis evidence is now overwhelming. At least 6 sites with reliable dates older than 13,000 years, supported by multiple methods and independent replication. The Bluefish Caves in the Yukon produced cut-marked horse and mammoth bones dating to 24,000 years ago, confirmed by electron microscopy as human-made. The SEM analysis was decisive. Natural processes, trampling, gnawing, geological abrasion, produced different mark patterns than stone tools. The bluefish marks match the stone tool signature exactly. The marks could not have been made by carnivore teeth, rodent gnawing, or geological processes. They were made by stone tools, wielded by humans who were processing megafauna in the Yukon 24,000 years ago. The picture that emerges is not of a single migration event, but of a long, complex, and gradual process. Humans entering Beringia from Northeast Asia over 20,000 years ago, occupying the land bridge for millennia, and gradually dispersing south through multiple routes and over multiple generations. The founding population was a mixture of ancient North Eurasian and East Asian ancestry, admixed in Beringia itself during the standstill. The emerging model is not one migration, but multiple. An early coastal route followed by later interior expansion once the ice-free corridor opened. Different routes, different times. Same founding population. The Beringian standstill, the founding population, lived on the land bridge for millennia. Genetically isolated, accumulating the mutations that define Native American lineages before dispersing south. Today, the Bering Strait is 85 kilometers of cold water. 20,000 years ago, it was grassland. The people who crossed it were not making a dramatic voyage. They were walking on dry ground, following animals and plants they already knew. The DNA evidence for early Americans is growing but still fragmentary. Ancient DNA from the earliest periods is rare. Hot, acidic, or waterlogged conditions in many American sites destroy genetic material faster than in the permafrost conditions that preserve Siberian DNA. The oldest American DNA comes from the driest and coldest sites: Paisley Caves in the Oregon Desert, Spirit Cave in Nevada, Upward Sunday River in Alaska. Tropical and eastern sites where many pre-Clovis occupations likely occurred yield almost no DNA. Teeth remain the most reliable source of ancient DNA in the Americas. The enamel shell protects the DNA-rich pulp cavity from the environmental degradation that destroys bone-based DNA. The distribution of ancient DNA recovery is uneven, concentrated where preservation conditions are best. The full story of the first Americans remains partially told, limited by the accidents of preservation. The Spirit Cave mummy from Nevada, over 10,000 years old, produced enough DNA for a full genome. The results showed clear affinity to modern Native American populations, with no European or other non-Asian ancestry. Every ancient American genome tells the same story: descent from the Northeast Asian founding population. No European migration, no Polynesian arrival. Singular origin. Diverse timing. The genetic unity is striking. Despite being separated by thousands of years and thousands of kilometers, every ancient American tested so far descends from the same founding population. A population that entered the Americas from Northeast Asia and diversified after arrival. The genetic continuity matters beyond academia. For indigenous communities, the DNA confirms what oral traditions have long maintained, that their ancestors have been in this land since time immemorial. The science is catching up to the stories. The first Americans were not a footnote in someone else's story. They were the founding population of 2 continents, and their descendants are still here, carrying the same DNA that walked beside that ancient lake 21,000 years ago. The textbooks are being rewritten. The question is no longer whether humans were in the Americas before Clovis. That debate is settled. The question now is how much earlier, and by what route. 21,000 years ago, in a land the textbook said was empty, someone walked beside a lake with their children. They left footprints in the mud became stone, and the stone waited 21 millennia to prove every textbook wrong. The footprints are still there, the DNA confirms it, and the first Americans were here long before anyone believed they could be.